Hello everyone. Today we are going to try to understand ATP. Let's start. Now, the first time you hear about ATP is it being called as the energy currency. So let's first talk about energy. How do you store energy? So energy on Earth is available as sun. Sun gives you all the energy, so it's available as light. But how do you store this energy? You use it to form chemical bonds. You use it to energize electrons and form bonds. What does that mean? Imagine two of your classmates who don't want to sit together. Now, what does your teacher do? Your teacher gives them a chocolate. Your teacher says if you sit together, they can get a chocolate. What is this chocolate in our terms? That is the energy. So energy can bring atoms together and let them stay together in a chemical bond, and now you can call them molecules. Plants or other bacteria that can capture the energy of light do exactly that. They convert the light energy and store it in big molecules like carbohydrates. Even methane, which has a lot of CH4 bonds, can store this, right? But for us, it is carbohydrate, a molecule which has a lot of CH and COH bonds. So the storage is complete, but how do we use this energy? Now, a very common misunderstanding is you can break these bonds to give you energy, but it is just half of the story. You remember, nobody likes to give away a chocolate, right? So in order to get them to release the energy, you need to invest some energy so that they can be broken, right? So you need to invest some energy to pull out that chocolate, and then you can have a net release of energy. So always you can have energy by breaking these compounds, but you will always be focused about a net release. Why a net release? Because you will eventually or initially invest some energy for breaking these. Now, why don't we simply use glucose to for all purposes of our cell? Simply because it's too much. We all have seen a dam. What is a dam? It stores a lot of water. But what were to happen if that dam breaks? Lot amount of energy would be released with the water and it would wreak havoc. And that will exactly happen if all amount of energy of the glucose, which is 686 kilocalories per mole, is released in a single step. So what do you do? The same thing that we do with dams. We release the water gradually in small pockets and then you make the electricity. Same way, you can't use glucose every now and then. It's too risky. So you use glucose to create energy, energy in to store energy in smaller pockets that you call as ATP, adenosine triphosphate. So, and then you break this ATP, adenosine triphosphate, three phosphates, with the help of water, hydrolysis, always hydrolysis because you break it. Lysis means you are breaking. Hydro means in presence of water. You're breaking this and you're pulling one phosphate apart, and that is the one thing that is causing a net release. 7.3 kilocalorie. In most of the textbooks, you'll see a minus sign written after that because we want to signify that the energy is being released. So let us once see the structure of ATP just to make things more clear for us. So ATP has a sugar, a base, and three phosphates. So the first is adenine, which is a base. The second is ribose, a sugar. This combined, you call them adenosine. Adenosine is adenine plus the sugar ribose. And then you have three phosphates. So when you break these bonds, so if you just break the last bond, you will have ADP plus phosphate and a release of 7.3 kilocalories per mole. Remember, one mole has 6.03 into 10 to the power 23 of these molecules. Now, how does that work? So adenosine triphosphate breaks. You have two possible ways to use that energy. The first one is directly. Imagine you have a plasma membrane, and in that plasma membrane you have a protein channel. So there is a protein channel that can allow something to go from one side to other or pull something from one side to other. The problem is something that needs to be pulled is simply not fitting. Let's say it's sodium, for example. So the release of this energy from the hydrolysis of ATP can cause conformational changes, changes in conformation, the fitting of this protein that we are talking about, and the sodium is allowed to be passed. So that's the direct release of energy. And using that direct energy release to cause conformational changes. The other way is coupling. What is coupling? Let us try to understand that. So I want to convert glutamic acid to glutamine. Now glutamine conversion requires it to combine with NH3. Alas, this reaction requires plus 3.4 kilocalorie per mole. Now what do I do is I use my ATP. So I break ATP. So I am gonna break ATP, and that is gonna give me ADP plus phosphate plus how much? I hope you remember 7.3. I'm gonna add a minus 7.3 so that you remember it's gonna be a release. This phosphate now binds with this glutamic acid. So remember, what was our goal? Our goal was to produce this reaction. Okay, that was your goal. Now I'm gonna use this phosphate to phosphorylate my glutamic acid. So this glut is gonna be phosphorylated for, so I'm just gonna write phosphorylated glutamate or glutamic acid. This is a phosphorylated intermediate. Now you will ask, what is the big deal about phosphorylation? The big deal is you have added a bond, and if you remember, bond means energy. I have increased the energy of the glutamate system. Now this phosphorylated glutamate reacts with NH3 to give you finally glutamine and the phosphate. So do you have what you wanted? Yes, you have glutamine. And did you get the energy that you needed? Yes. You wanted 3.4 kilocalorie, and that was available in the form of phosphorylation. But where is the remaining? So you wanted 3.4, you were given 7.3. If you do the maths, you have a total of minus 3.9. This is the energy that is going to be released. Any reaction will continue to take place as long as there is a net reduction in the energy of the system. So this 3.9 is given out, and now the net energy content of the system is less because the 3.9 has been released out. This is how you use coupling to drive one reaction that requires energy and another that gives off energy. In this case, that was the breaking hydrolysis of ATP. So let's summarize. So adenosine triphosphate can act as energy currency because of hydrolysis reaction. You can use the hydrolysis in two ways: directly, like by causing a conformational change, or by coupling it with another reaction. Thank you. I hope this was able to help you something to understand this with another reaction. Thank you. I hope this was able to help you something to understand this process.
Can't find a biology topic you're struggling with? Let us know in the comments section and subscribe to check out new videos.